Welcome to our series of virtual voices presented by United Nations World Information Transfer, focusing on the United Nations 17 Sustainable Development Goals. Our speaker today is Andre Watson, who is a scientist, inventor, and CEO of Ligandal, a precision genetic medicine company initially focused on hematology, immuno-oncology, rare genetic diseases, and infectious diseases. His mission is to create precision medicine that can address underlying diseases, states curatively, predictively, and efficiently. During his undergraduate research at Rensselaer Polytechnic Institute, he developed a gene editing delivery technology for shuttling technologies such as CRISPR and TALEN gene editors into specific cell types. He subsequently founded Ligandal in 2014 to commercialize cell tissue and organ-specific drug and gene delivery systems. He's an inventor on 59 pending patents globally across 14 patent families, and in 2021 was listed on Forbes 30 under 30 for healthcare. Over to you, Andre.
Hi, all. Thank you so much for the opportunity to share what we are building with you all today. I'm just gonna pull up a few slides here. So really the purpose of the presentation today is to talk about precision medicine and what this means for not just the future of healthcare, but how these medicines can be increasingly affordable and accessible. Can everyone see my screen? So to start, Ligandal, really what we are creating is, as Mariam mentioned, cell, organ, and tissue specific delivery technologies and targeting technologies for a variety of drugs, and particularly we've had a focus on gene therapies. So taking technologies that can predictively and curatively address underlying disease states, especially for rare genetic diseases where you have an underlying mutation that drives the disease. If you can correct that underlying mutation, you no longer have the disease. So therefore, a lot of our interest is in how do we shuttle these next generation gene editing technologies, as well as mRNA therapies and DNA technologies, into specific cell types and tissues in order to cure the diseases. So I don't think, well, here. So precision medicine is really the notion of tailoring a therapy to an individual, and under precision medicine, you also have personalized medicine. So you can think of this in two ways. With precision medicine, it could be as simple as you have a disease, it's in, let's say, your kidneys, and you would like to address the delivery to the kidney, but the drug that you want to deliver might go everywhere or not go to the kidney at all. So if you can tailor that to the individual, then you increase the precision, the efficacy, and potentially the safety of a drug. Now, in the case of gene therapies, this is very interesting because gene therapies don't target any tissue necessarily. For the most part, the delivery systems that are used for these gene therapies are going to the liver, and targeting them can be quite difficult. So where we're interested is how do we address the precision medicine aspect of this? There are more than 10,000 rare genetic diseases encompassing over 2.2 million mutations that are known. And there are some that are more common than others, and they have varying prevalence in developing nations as well. And we'll talk a little bit more about this later. But for instance, in North Africa, up to 40% of the population are carriers for at least one copy of sickle cell disease or beta thalassemia. And about 2% of the population or so has one of these diseases or the other. So if you can actually curatively address that, you're looking at something that can affect millions of lives. Now, the personalized medicine aspect says we might not even have a drug to treat a person, but we know what's wrong with them, and designing the actual therapeutic that can address that. So right now, a lot of these therapies are very expensive, but where we see this going is that there's going to be a significant drop in cost in the next 10 to 20 years as you sort of see these mainstream initial rare diseases go through the FDA, get approvals. They may initially cost millions of dollars, as we're seeing in the United States. But once you can apply these technologies to thousands of diseases, they don't need to cost as much. So it's really a volume issue. And we've already seen that to some extent with mRNA technologies, companies like Moderna and Pfizer, BioNTech, in commercializing these nanoparticle-based mRNA delivery systems for vaccine technology. Now, they're delivering a very small dose, so you can do that for about $20 per person. Gene therapy, you typically would need a much larger dose. So the cost of goods, if you're administering to hundreds of millions or billions of people, may be closer to tens of thousands of dollars with current technologies. But once this starts dropping to around that range or lower, you can really start to see how these technologies can be widely distributed for not just vaccine applications, as we're seeing right now, but really for underlying disease pathologies for thousands of diseases. So really where this plays into the UN's Sustainable Development Goals is the good health and well-being, industry, innovation, and infrastructure, reducing inequalities, and particularly in relation to health equity. We see a lot of opportunity to collaborate with many different research institutes, academic groups, pharmaceutical companies, and even governments. And we'll talk a little bit more about the specific applications that are of interest in the short term to long term. So really, as I've already mentioned, creating affordable treatments and accessible treatments is critical for global health equity. You can't just have these multi-million-dollar drugs and gene therapies, or gene therapy specifically. Looking at, for instance, the first gene therapy approved for beta thalassemia in the United States, it costs $2.7 million per person, and it's supposed to be once and done, but this isn't really practical for a global accessibility from a global accessibility perspective. So creating something that can be increasingly personalized and precise, and where the drug itself is evolving to the individual patient's needs. So perhaps you have a technology that can target the liver or the kidney, but you want to target the lungs or the spleen. Where we focus is really on how to change the targeting and tailor that delivery to the specific organ or tissue that needs the therapeutic. And in the case of mRNA therapies, CRISPR technologies, all these gene editing technologies, and some of these more advanced genetic medicine interventions, you can tailor not just where you're targeting it, but you can also change the sequence that you're delivering, or the sequence that you're changing in the person's DNA, or the mRNA sequence that you want to transiently produce in the patient. So if you go from one organ to a different organ and you change the sequence on the inside, you're not looking at going through the typical drug development process of having to discover a new molecule from scratch, essentially screening thousands or looking at potentially millions of potential molecules. You're actually knowing predictively that this is a mutation that the person has, and that's why they have the disease. So I really foresee that there's going to be a vast acceleration of the development of these therapeutics and their personalization and precision in the coming years and decades. So the long-term impact to patients is really creating precision medicine that can improve patient outcomes and changing a lot of the paradigm of how drugs are developed and the effects that they have from being something that's palliative, or that you need to continuously take and that relieves your symptoms, but doesn't necessarily address the underlying disease pathology. Shifting that paradigm to one of predictive and personalized and actually curative, where you know that your intervention is actually addressing the underlying disease state. Our model as a company is really that we are partnering with other pharmaceutical companies to enable them to lead the commercialization efforts in the clinical trials. So we're a discovery company that licenses specific products and technologies to other companies to create enhancements to or novel therapeutics around specific disease areas. And you can think about how, for instance, going back to mRNA technologies, right now there's a pretty limited use case for mRNA technologies because of the limited targeting to different organs and tissues. Same thing with CRISPR technologies, gene editing technologies, DNA therapies, and all of this. So if you can actually address the targeting to any number of organs and tissues predictively and across multiple disease states, you're looking at expanding the addressable diseases potentially hundreds of fold or even thousands of fold. And that's really what our mission is, and to create an ecosystem of technology coupled with commercializing entities for these specific indications. And here are some specific examples of some rare hematological rare diseases. And these are some of our initial focus areas. The interesting part of this is that it's all centered around one cell type, or hematopoietic stem cell. So what you're looking at here is, in the case of sickle cell, it's one letter of DNA that is messed up. And if you can correct that letter, you no longer have the disease. The issue is, how do you correct this letter? And so right now, a lot of the therapeutics in development around this are taking cells out of the body, and they're engineering them and then putting them back in with the correct mutation. This is very expensive. It costs hundreds of thousands of dollars just to manufacture. And the drugs, both for sickle cell and beta thalassemia, in the case of Bluebird Bio, their drug is $2.7 million, as I mentioned earlier. So in this case, if you can target the underlying cell type, the hematopoietic stem cell, which resides in your bone marrow and sometimes circulates in your blood, if you can correct a certain percentage of those cells and actually fix that mutation, you would no longer have the disease. And so a lot of our focus is not just on how do we provide enhanced technologies for cell therapy companies, but also developing the in vivo targeting technologies, so you never have to take cells out of the body to begin with. So we are wrapping the genetic material, or the gene editors, into these nanoparticles that then have a targeting molecule on the surface that can bind to the specific marker that is found on the cell type that you want to target. So essentially, you get these separate degrees of freedom. You don't need one drug to perform the effect that you want and go to the right place. You're separating the targeting of the where from the what are you doing and the actual drug that is delivered to the place of interest. HIV is also an interesting example, because in this case, this virus actually edits your immune cells to insert itself permanently. And if you can actually edit the same cells that are affected by sickle cell and beta thalassemia, you can actually confer a resistance to HIV. It's this mutation that Magic Johnson had famously, where you can have this receptor that HIV enters a cell through and mutate it in such a way that HIV can no longer enter. So if you look at a place like Africa as a whole, in Northern Africa in particular, with a sickle cell and beta thalassemia prevalence, if you can create a targeting technology that's affordable and accessible for that cell type, you're looking at something that could apply to all three of these diseases. And so the other big point of interest for us is biodefense and emerging infectious diseases, because we realized that our technology during the COVID pandemic could be applied to not just targeting cell types, but actually mimicking how pathogens target cells. And in doing so, we created a sort of Trojan horse approach, where we synthesize these synthetic peptides, which are small protein fragments of larger proteins that we can chemically make very rapidly. And we tailored these, in the case of COVID, to replicate the part of the virus that sticks to the cell. So it sort of acts like a Trojan horse, binding to the same part of the cell as a virus binds to, but inhibiting the virus from being able to enter. And this can also play a role in future vaccine technologies. So a big part of our focus is in how do we create infectious disease mitigating agents that can also be predictively modeled and rapidly created and act as both prophylactics and therapeutics, with potential utility as vaccine technologies. So that's really the focus of the presentation here today. But beyond that, can talk a little bit more about the technology and the background of all of this. So really what we do is we've built out a large database of all of the markers, all of the genetic mutations that exist, and how these markers are broken down organ by organ, tissue by tissue, and cell by cell. subsequently, we have an understanding of how your natural biology interacts with those various markers. So we have an ability to look at what we call protein-protein interactions, or in the case of the markers, what naturally is binding to them in your own biology. And using three-dimensional modeling and AI techniques, we're able to sort of reverse engineer those interactions and clip out the parts of those interactions that we want and replicate them into synthetic sequences of strings of amino acids, which you would find in a protein that we can make fully synthetically. As a result of that, we rapidly reduce the screening time and increase the predictive power of these algorithms so that when we actually synthesize a molecule, we're making 5 or 10 things, not a trillion different sequences of random sequences of peptides in this case. And then the ability to apply that targeting technology to various modalities, ranging from, if you say a classical example would be, you have a chemotherapy drug. It has off-target toxicity. You really wanted to reach the cancer cell. You know what markers the cancer cells have. So what we're able to do is create a targeting peptide for that specific cancer cell or set of cells and tether it to a chemotherapy agent. That would be one example. I mentioned all the rare disease applications with genetic therapies. Another example would be that you have some drug that works pretty well. It has FDA approval, but it has toxicity in certain organs where you don't want it to go. So being able to direct more of that drug to the tissues and organs that you do want to target without targeting the organs that you don't want to target. So with that, I think I ran through this pretty quickly here. So can probably start asking questions.
I have a question. Can you talk a little bit more about your business model? Like how many patents do you guys have? How much of those can be licensed? How many, how much of that is currently being licensed, and for what amount? Love to better understand that.
Yeah, sure. So our patent portfolio is 59 patents across 14 families. It's been in development for about 10 years. So what it really encompasses is everything that I mentioned to you. One is sort of the database and AI-based approaches for generating these molecules to target specific markers on specific cells. And we have knowledge of all the markers. So it's really an agnostic approach in that sense. The second is the delivery technologies. So I've been working on drug and gene delivery technologies for about 12, almost 13 years now. Originally working with gene editing technologies, but it really expanded when we started the company. So a lot of our patents focus on the compositions of matter around the actual nanoparticles and the delivery systems that could be used for different technologies, ranging from mRNA to CRISPR technologies, TALENs, which is another gene editing tool, and also DNA, silencing RNA, and proteins. And also we can apply this to small molecules. Finally, a lot of the patents focus on the specific sequences of peptides that we generate. So we'll have broad patents around this sort of method and the use cases and how this can be applied algorithmically and predictively. But then each of those patents spins off into specific sequences for specific cell types with the specific disease areas and payloads, the mRNA, DNA, et cetera, that can be delivered, also encompassed in those patents. So it's very broad. I like to think of it as sort of having 3 components: the targeting, the delivery, and then the active molecule that needs to be in the cells. In terms of what we're currently licensing out, I can't really talk about that in a public setting or the economics of our deals.
Thank you.
Any other questions?
Thank you. We've received a few questions from our audience, actually, so I can read some of those. The first one being listed on Forbes 30 under 30 for healthcare in 2021 is a significant achievement. So how do you envision your work impacting the healthcare industry in the future?
Yeah, right now it can cost billions of dollars to develop a drug, and the failure rate is very high. And this means that by the time you've developed a drug, you've spent so much money that frequently that drug has to be very expensive. Unless it can really address a very large population, then you can see a drop in the cost. In the case of gene therapies and rare diseases, there's sort of this conflict from a development perspective in that, let's say you want to create a therapy for an ultra rare disease and only 200 people in the world have that disease. You will still spend tens or hundreds of millions of dollars to develop something to treat it. So ultimately, when it gets approval, you have to price it in the millions of dollars or high hundreds of thousands of dollars to even begin recouping the cost of development. And this has to do with this sort of older paradigm of the drug taking a very long time to develop, being very expensive, and typically only being applied to one disease or a small set of diseases. Where we are seeing things going, and this is also based on some conversations I had with the head of the biologics division of the FDA. I posed this question of, well, let's say I have an mRNA technology, like say you take Moderna as a great example of this. We know that it's been applied to X disease. It costs a certain amount to develop. At what point, A, is there comfort in changing the mRNA sequence and applying it to a different disease? And B, changing the targeting of the ultimate delivery system to go to a different organ with something that has already gone through safety and efficacy studies for a different organ and essentially fast tracking that. So the answer was quite interesting in that there's this notion of a new sort of regulatory pathway, or a newer one called a bio-better, which is that you're changing the drug, or in this case, the delivery system, or the mRNA sequence, or the targeting to encompass a new application or set of applications. And there's also the possibility of engaging what is being called basket clinical trials. So you could say, I can target kidney, and here are 30 genetic diseases in the kidney. And as you get the preliminary safety and efficacy studies for, let's say, one disease, you can actually roll in multiple other diseases into the same trial. And you end up having a platform that can be applied to many things. So what really the long-term vision here is that as we unlock the targeting for all of the organs, tissues, and cell types in the body that are relevant to these disease states, those become validated technologies. And then those technologies can be married to different technologies, including mRNA, DNA, CRISPR technologies, silencing RNA therapies, other next generation gene specific technologies, and even small molecules, repurposing them or enhancing their efficacy. So once you get the initial legwork done of the approvals for these rare diseases, for the lethal diseases, for the cancers, for the really unaddressed or under-addressed diseases, you can start to expand your patient populations with these technologies to ultimately go after things that are not rare anymore. And so where I would like to see medicine go is that in 20 years, if you have a disease of some kind, you're actually able to not just target the specific cells and the specific disease pathology, but cure it and or address the underlying disease in at the most basal level of what is driving the disease, as opposed to something downstream. And that this can be broadly enough accessible and implemented in the healthcare markets, as well as used by a number of pharmaceutical companies, such that your doctor's job is really just to figure out the diagnostic element, and then that gets coupled almost automatically to a specific therapeutic embodiment that is tailored to just you or to people with your very specific disease.
Sounds amazing. And do you see any opportunities for global implementation of your technology to achieve this path in the near term?
Yeah, I mean, look at it depends on what you define as near term. I mean, I think it will be a multi-pronged effort that will require engagement of different governments, NGOs, and whatnot. But I really like the Africa example, because that's an obvious use case for a rare genetic disease in the US that is actually more prevalent in Africa, and that ultimately we know exactly what's wrong. We know exactly what we need to fix. And the problem is just that the technologies necessary to fix that are currently being priced in the millions of dollars in the United States. But there's no reason fundamentally that as these technologies are developed, we can't manufacture them somewhere on the order of low to mid tens of thousands of dollars per patient. And given that these would be curative implementations, I don't think that that is necessarily unreasonable. So how that looks going forward 10 years or 15 years or 20 years, I think we're going to see further drops in cost. So for instance, the peptide technologies that we are using are pretty cheap to actually manufacture. They could be rapidly manufactured, and they can be manufactured in a distributed way. So a big part of where we're interested is also how to address sort of distributed manufacturing, so that you don't just have to manufacture everything in one facility and figure out how to ship it. You can actually have different centers and manufacturing plants that can be miniaturized across the globe and applied to the specific disease areas that are necessary for a given hospital, a given area, or a given country even. So I think this will get affordable and accessible. It's just a matter of you sort of need that initial wedge into the US regulatory market for a lot of these technologies to really succeed. But as you're making progress, there's no reason not to engage other countries as well and try to drop the cost or provide at cost or nearly at cost, so that you're essentially subsidizing some of those cost bases with either NGO money, local government money, and or even some of our proceeds from US and developed nations.
What are the main challenges you foresee with implementing precision targeted medicine and ways to potentially combat those?
I think a lot of it is regulatory, and I think a lot of it is sort of around the ethics of how do you implement clinical trials or tests on these things. In the rare disease context, it's a little more obvious that you could do faster clinical trials. You could pilot things and see how well they are working, especially when you know exactly what's wrong and where you need to go to fix it. But ultimately, a lot of these studies still require going through animal models, humanized animal models, monkeys, and then humans. And I think the goal is to shift this to a point where you don't need to do all of that. But in order to get to that point, we still need to perform the studies and ultimately get the initial clinical validation for each of the relevant organs and tissues. Once we pass that point, which, like I said, would probably be in a developed nation initially, but it would be with some of these rare diseases affecting each of those organs, I think there's a good case to be made for applying that to larger population sets in developing nations. The goal is just not to, treat developing nations as sort of guinea pigs for technology that has risks. It needs to be really well put together in a clear regulatory format that has strong ethics for diseases that are under-addressed, lethal, or so rare that there are no interventions. And then once you have the strong enough data set that is safe and effective, then yes, expanding that is, I think, relatively easy with the right government backing and financial backing. So.
Thank you. And your response ties into the following question, which is around whether there are ethical concerns associated with personalized medicine and how they can be addressed. Anything else to add on this topic?
Yeah, I mean, I think most of the ethics are around, there are several points of ethics to consider. One is with gene therapies, are there unintended consequences or side effects? So whenever you're dealing with gene editors, you're usually introducing unwanted mutations as well. And there's sort of an acceptable level of risk of doing that with a rare disease, but also if you're going to go give something to millions of people, you really want to know that your technology is not causing higher rates of cancer long-term or unwanted mutations. The good news is that you're not getting mutations that are what we call germline. So it's not editing the testes or ovaries. So you're not looking at having something that can be passed on. So with these rare disease cases, if you have diseases where the expected lifespan is 35 or 40 years old, you have to weigh that out. I think those technologies are improving. I think we are going to get to a point where we can do pretty much seamless gene insertions and deletions, and that might be 5 or 10 years away. But once those technologies are developed and honed in, then that ethics question becomes less of a concern. So for instance, using first generation CRISPR technologies, which are really, they're cutting, they're not pasting. Every time you cut DNA and you just create a double-stranded break in the DNA, you're creating random insertions and deletions around that sequence, which can cause unintended consequences. The other parts are, as I mentioned a bit before, really around choosing the disease indications, and how that applies to further non-rare diseases is also of ethical concern, and that really ties to the safety of the compound that you're delivering, and in some cases also the delivery system. So in the case of Moderna, BioNTech, Pfizer's delivery technologies for mRNA, they're quite toxic at higher doses, and the actual lipid technology used to package the mRNA is problematic when you're going into repeat dosing for 1,000 times a vaccine dose, for instance. So as you go and administer this, in the case of mRNA, every 2 weeks for years on end for a patient, you're looking at potential safety concerns around the actual delivery technology. So you really need to assess all these things, and that is part of the regulatory process, that you have to do all the absorption, distribution, metabolism, elimination, and toxicology studies, not just for the drug, but also the carrier of the drug if relevant. And so it may take time to see these technologies implemented at a global scale for non-rare diseases and non-lethal diseases, and outside of this vaccine vertical where the dose is very small, chiefly due to those concerns. So.
Interesting. Are there any cultural or societal factors that affect the adoption of precision medicine in different regions in the world?
I mean, I'm sure that there are different cultures that view naturopathic medicine as probably something that is appropriate, and anything that is like genetic or otherwise is not. And I mean, if you go to a tribe somewhere, they might say this is some sort of voodoo magic that you're doing. We don't want it. So I think that's definitely something that needs to be considered as well. And I think addressing also that these are not germline edits that get passed on to future generations is also important. I mean, we're not looking at doing embryological gene editing here. That's a whole Pandora's box of ethical concerns that we're not looking to deal with. So.
Has your work been impacted by the COVID-19 pandemic, and are you aware of how COVID affects the work of scientists in creating and manufacturing medicines around the world?
Yeah, COVID was really tough because there was just this complete refocusing on biodefense, and realizing that there could be what you could call a zero notice pathogen that appears out of nowhere and suddenly propagates everywhere. So looking at COVID, we actually focused a lot of our technology on, could we apply this to COVID? And we designed a peptide that could block the virus from entering the cells, that we partnered with the National Institutes of Health on, and so far has been 99.7% effective in reducing the amount of virus present. So it was both a terrible thing, and from a technology innovation and regulatory acceleration perspective, it was a good thing for these platform technologies. So terrible for humanity, but very good from perspective of now we have mRNA technologies in humans. So if you had asked me in 2019, how long will it take for 1 billion doses of mRNA non-viral gene therapy technologies, and in the US, they don't like to call it gene therapy because it's a vaccine and it's a small dose of mRNA. In Europe, they do like to call it gene therapy. So I'm just going to lay that out. But these are gene therapy technologies. They're just not permanent changes that are being made. I would have told you in 2019 that it would probably take 10 to 20 years or 10 to 15 years. And so seeing that happen in a matter of a year or two was unexpected and fascinating from a perspective that now we have FDA approved mRNA technologies that can now be applied to all of these other diseases. So seeing that and how that is going to evolve in the coming years, I think it's enabled a lot of innovation outside of COVID that is going to be paying dividends in the next 5 or 10 years, with respect to all the lives that were lost and how terrible the pandemic was.
What led you to pursue this mission and field?
I was initially really interested in physics and actually music. I decided that I was initially very interested in neuroscience and understanding how the brain works, but I decided that ultimately biomedical engineering was the area I wanted to focus on. And within that, drug and gene therapy and gene delivery really struck me as where I would like to focus, because I realized that we had, a complete understanding of the human genome sequence, for instance. We had an understanding of thousands of diseases and what is actually driving them. And then looking at what the interventions were for those diseases, there were so few. And ultimately, applying these technologies, the knowledge of what is wrong with the ability to fix it, sort of that molecular biology toolbox that we now have to delete, insert, cut, paste, turn off, turn on, or temporarily produce any genetic sequence we want. The question was, why isn't this being applied to all of these diseases? And so the answer was really delivery, that if you could deliver efficaciously to the different organs and tissues and couple that to these modalities, you could be addressing hundreds of thousands of more fold diseases than are currently being addressed at the genetic level. So I felt that that was the most impactful thing that I could focus on for the next 20 years when I was starting this research, and was also very interested in the notion of regenerative medicine and how this plays a role in once you've established that you can apply this to the rare diseases, the lethal diseases, the more prevalent diseases, you have applications in regenerative medicine. So actually looking at you have an aging organ and you want to reduce its biological age or create more stem cells directly in the organ or kill off aged and diseased cells. So coupling this sort of technology with those applications, I saw that this could be something that could not just extend the lifespan of people, but also potentially impact the health span, so that by the time you're 80 or 90 years old, you don't have to biologically be 80 or 90 years old. You might be able to reduce that biological age. And, going after these aging interventions as an initial focus area isn't really possible because we don't define aging as a disease. So looking at how to apply the initial diseases that I mentioned and disease areas that I mentioned as sort of the stepping stone to eventually being able to do that is something that I'm very interested in.
What advice would you give to aspiring scientists and entrepreneurs looking to make a significant impact in the healthcare space?
I would say if you really care about one disease, focus on that, and that's great. But if you can create platforms that can be applied across multiple disease areas, you'll have probably more fulfillment in your development and in your process of working on this, because it's not going to be as fast as you would like it to be. If you're 20 years old and you're starting a company or thinking about starting a company, you might think you can accomplish a certain amount in 3 to 5 years. And it'll probably take you 7 to 10 years, but you'll probably accomplish more in 10 to 15 years than you thought you would, and probably less in 3 to 5 years than you thought you would. So it's you have to have a lot of patience, and it's not always easy for not the reasons you think it's not going to be easy. Sometimes the technology and the science is the easier part. You have to gather resources around it. You have to align people, and it really, if you want to have access to building something outside of an academic environment, for instance, it really does become a lot about people and how you manage your relationships and all of that. And you need to have a great team. So that can't be underscored enough.
What are some of the key challenges you've faced in your career, and how have you overcome them?
When I first started the company, I was 22. I think I had developed this proof of concept in an academic setting. I really wanted to spin it out. I came out to Silicon Valley. Initially, I had no resources at all, so I really had to figure out how to network and meet people and get the resources that we needed. That took a number of years. I thought that process would have been easier, but it took about 3 years to raise a seed round. And then with that, we were able to deploy a lot of resources. I think some of the other challenges have been aligning sort of the long-term impact of what we're doing with the the timelines that some venture capital firms would want to have around, well, you could just sell the company in 2 to 3 years. Why wouldn't you take that M&A offer? And so seeing how sort of impact can misalign with short-term financial incentives, that's probably been one of the things that has required working through and persistence, because not everyone who can write you a check is necessarily the right person to write you a check if you're working on something that takes 5, 10, or 15 years. So if you're building something that could be, sold in 3 years and that's the goal, then that's great. But I think learning that whole ecosystem and really finding the right shareholders, some of which are on the call today, that's been both the challenging process, but also really rewarding when you meet the right people. It's amazing. So.
And what do you think are the most effective ways to establish trust with customers and other stakeholders when you're starting a business?
I think you need a baseline of data. You need a baseline of having a minimum amount of resources, and you need to be quite persistent. And sometimes what is initially a no turns into a yes years later. So you really need to just have initially, you just need to have the confidence in what you are creating and the plans and the contingency plans for, if this given experiment doesn't work, what are you going to do around it? So it's always not just planning for here's what's going to work and I'm building something that I believe will work, but also, okay, if this particular experiment doesn't work, what is the pivot around that to create something that does? So you always need to be thinking about failure modes, especially in a context of partnering, whether it's academic or industry or otherwise. And if you're building an advanced and novel technology that is a platform, you will have things that don't work. So you need to sort of have the faith in your own your team, your technology, the platform, and the persistence around it, more so than on any single partnership or application, I think.
What do you think are the main outstanding areas that need to be studied in order to create and effectively implement individualized treatment?
I think at some point we will have an idea pretty soon of how to target all of the human organs and tissues. That I'm very confident in. The next piece will be sort of triaging the diseases according to how easy they are to address with existing technologies. So if you want to temporarily turn off a gene or temporarily introduce a gene, that's pretty easy. If you want to cut out a gene, that's pretty easy. If you want to insert a gene, that's still very hard. So if you want to permanently insert a gene or replace a gene segment, that is still very hard to do in the body because of all the safety concerns we mentioned earlier. So I think that in terms of creating personalized treatments at the genetic level, those are kind of the orders of difficulty that we'll also see correspondingly in how distributed they are in the market. In terms of drugs, there are a lot of drugs that could be either repurposed or that already work for a given application but could be better. And so I think there's a pretty good knowledge of what those drugs are, what their modalities and methods of actions are, and where they need to go. So this is really a resources issue more than a technological issue in a sense, and a regulatory issue. So there needs to be some long-term incentive for creating precision and personalized medicine, and some initiatives around how to ethically do those clinical trials, and sort of thinking outside of the box while not stepping on the toes of regulators who ultimately are going to be the decision makers about a lot of these things going to market. So there is sort of this push and pull with innovation and regulation, and you need some regulation and you need innovation, but if you go too innovative, you can surpass what is ethically or regulatorily reasonable. So there is also, I think, a bit of a convergence path for the point when the technologies are sufficiently mature, as I mentioned, with the rare, the lethal diseases, and applying then those same technologies or platforms to the other applications that are not rare. So I think it's more of a matter of time, money, and regulatory paths opening up than it is about a technological missing piece with the exception of the precision gene insertion technologies, which I really don't believe exist yet. So
Makes sense. Thank you.
Susie, if you don't mind, there is a question that came up, and I don't know if the person, he's actually an investor in these types of things, and he is one of the people in the audience, and he was asking if you could talk about the business itself, Andre, you know, how you evolved with the business and how it's working, and yeah, basically what the business is like for you now and how you're going to deal with it.
Yeah, so the business right now is that I feel we've established a strong IP moat. We just brought on Tushar Newell, who's on the call here, as our COO. He previously was head of business development and licensing for Novartis' Sandoz division, and has done about $2 billion worth of biopharma transactions in his career at companies ranging from Pfizer to Novartis and others. So I think now with all of the work that we've done in building the platform, building the intellectual property portfolio, validating the initial scientific forays, and showing that we can target different cells like immune cells, rare blood cells, bone marrow, and also applying this technology to the infectious disease applications. Really, the next focus of us as we enter our Series A is going to be to partner with one or more pharma companies to license out those initial sets of applications for anywhere from 3 to 9 diseases per partnership. And the deal dynamics are that pharma is really good at distribution. They're very good at commercialization and clinical trials. Over the last few decades, they've become progressively worse at innovation. So the most recent internal rates of return are below inflation. So for every R&D dollar spent by a pharma company, they're actually losing money. So they require, forgive me, they require licensing technologies from smaller biotech companies in order to build the portfolios of products that they need to continue having a competitive edge as we enter an era of more personalized medicine, gene therapies, complex biologic therapies, and so on and so forth. So our business right now is we have one biotech deal in place. We have one government partnership and we have an academic partnership. But now we're pursuing other pharmaceutical partnerships that, if you look at the deal dynamics, according to the most recent JP Morgan data, the median upfront deal value, so a pharma company paying to a smaller biotech, is $42 million for the discovery/platform stage, going up to $60 million upfront at the later stage preclinical/IND enabling, so investigative new drug enabling study stage, which goes up to just north of $100 million between phase 1 and 2, and then drops a little bit at phase 3. So for each of those partnerships, you also get milestone payments for each of the drugs or disease areas that are licensed from your portfolio of patents. So for any given disease, you'll get $200 to $300 million in milestone payments for each disease. And typically they're licensing anywhere from 3 at the very low end up to 9 or so at the high end. So you get these deal values that are in the billions. And so what we're pursuing is, how do we, rather than spending tens or hundreds of millions of dollars to get one drug into clinical trials ourselves and then having all the risk, we are actually distributing that across pharmaceutical partnerships so we can bring in the revenue from those upfronts and those milestone payments and get the validation from a clinical perspective and handle have the pharmaceutical companies handling the distribution and the clinical studies and the ultimate all the post-market regulatory aspects of those disease areas, while we also retain a lot of technology for corresponding disease areas that don't overlap with what that partnership would be around. So that's really how we're thinking about it. So our goals over the next year or so, maybe year and a half, are to close our first big pharma deal and then expand to additional ones for additional organ systems and disease areas.
Can you also tell us how long it takes for these patents to be basically secured? And
yeah, well, we file a lot of IP. So our first patent took years to get approval, but that was filed in 2013. And then we've been filing consistently a lot of patents over the years. It can take anywhere from a couple years after initial filing to 5 years or more to get your final office actions, and then it's a back and forth with the examiner for each of the countries that you're dealing with. So you definitely need to have a very good IP lawyer. We work with Michael Weiss at Perkins Coie, who won the CRISPR case for MIT, Editas, and Broad versus UC Berkeley. So it's a process. So you have to be kind of balanced between being preemptive enough around what you want to do without sort of acting as your own strongest prior art as well. So sometimes we'll file a patent and a couple of claims will get rejected on the basis of a prior patent that we filed. So you're like your own worst enemy if you're too innovative. But if you're not innovative enough, then someone else will come along and try to file the claim. So
interesting. So so tell me about your focus and your concept of how the United Nations can help you, because I know you've shown a recent interest in how you would like to sort of transmit this information there. So tell us how we can help in that.
Yeah, I think short term, I'm very interested in the emerging infectious disease angle. So creating room temperature stable compounds that can mitigate, prevent, and even act as vaccine technologies for novel or emerging viral agents that are popping up in different areas, especially as we see climate change take hold and there being more flooding, thawing of permafrost, and all of these environmental effects. There's correspondingly an increase in the presence of certain pathogens and their emergence, not to mention novel pathogens, not to mention things that aren't even natural, that are a risk that could be released by bad actors. So a lot of our interest is in how do you go from identifying the sequence of a pathogen to creating something that blocks it in a matter of days or weeks, and create something that can be readily distributed to the areas that need it, to also prevent transmission and reduce the severity of the disease. So that's something that we're very interested in from a perspective of gaining a greater knowledge of and connection with multiple governments in order to bring these technologies forward. Then there's also the specific disease areas that have specific prevalences in different countries. So the hematology aspect is really interesting in Northern Africa, for instance, as well as in the Gulf, in the Middle East. It's very prevalent. You have a lot of sickle cell, a lot of beta thalassemia. So it's not necessarily just a personalized or precision medicine approach for individuals. It's also something that can be tailored for different countries, and creating initiatives around maybe we want to have an initiative that in the next 10 years we can address greater than a certain percentage of sickle cell, beta thalassemia, and maybe a few other diseases and their prevalences in given countries. So I'm very interested in that aspect, because this isn't just a US thing. I mean, the whole world needs this technology.
Susie, is there any questions that you have?
Yes, there are a couple more left from our audience questions. So could precision medicine slow down the treatment of patients when providing customized solutions? And if so, how could this be mitigated?
Could it slow down the access or the providing of treatments to? Okay, it's an interesting question. I mean, if it's cost prohibitive, then it could slow it down. The question is, what is the comparison? Because anytime you go and develop a drug and you go through the FDA, you're always being benchmarked against what the standard of care is. So if there is a standard of care and it's accessible, then nothing's preventing you from getting that except for maybe the cost and getting insurance or otherwise, or maybe certain countries don't have access to certain medicines as much. But then when you go personalized, it could initially be very expensive. Absolutely. I think initially, just looking at right now, like I mentioned, beta thalassemia is costing $2.7 million in the US for a gene therapy that's supposed to be once and done. So, is it possible that initially most of these things will cost hundreds of thousands of dollars or millions of dollars? I think it's absolutely possible. I think there is potentially a gap there where for some period of time, certain diseases and certain applications might be inaccessible. And that's challenging from a perspective of, well, if you don't go after those diseases, then you'll never have a therapy for it. If you don't charge enough initially, then you'll never recoup the costs on the development that you spent. So no one gets the treatment. And at the same time, there's this balance with, if you have a drug that's making hundreds of millions or billions of dollars, but a certain country can't afford it, I think it's more about how do you sponsor and finance those disease areas. And yeah, I mean, it's an interesting question. I'll have to think about it a little more because there's so many incentives involved in all of this that there could even be a couple of gaps that I haven't thought of there.
Thank you. And what role does data sharing and collaboration among healthcare institutions and researchers play in advancing this type of medicine forward?
Yeah, I mean, right now, electronic health records are kind of a mess between hospitals. So if you have data from one hospital and you want to send it to another hospital or aggregate that data, it can be very challenging to do that. The good news is that when it comes to genetic diseases, there are pretty good existing databases and data sets that are hosted by the US government, for instance, that can be accessed by anyone. And they're not tied to any individual person. They're just saying this disease has these mutations. So initially looking at that and figuring out the prevalences of those diseases, that's an exercise that's pretty reasonable. But then to actually be able to go to a hospital system and keep in mind there's also a need for more advanced and preemptive diagnostics. So when you go to a doctor, you're not typically getting your whole genome sequenced. You're not typically getting, if you have cancer, you're not getting a biopsy with, you'll get certain DNA mutations associated with that cancer, but you're not getting proteomic scans or screens, and you're not getting single cell RNA sequencing screens that establish what the markers are beyond at the high end, looking at 750 or so mutations. So I think some of the paradigms around diagnostics need to shift as well. And we need increasingly advanced diagnostics and even theranostics where you might be able to track how something will go to the intended target before you administer a larger dose of it. So that's all something that I think tying all of that to hospital ecosystems and even nationwide or statewide or worldwide coverage for increasingly personalized needs, there are going to be a lot of overhauls that need to happen and a lot of advancements that need to happen on the diagnostics front in order to see this really be the standard of care, I would say.
Thank you. In our last minute, what are the best ways participants of this webinar can learn more and get further involved in your work? And is there a place to donate?
Well, we're a private company, so we have a number of accredited investor paths to for interested parties where we have the capability of discussing that. We're not doing a public fundraise, so I can't solicit capital on a call, but you're welcome to reach out to me if you're interested in learning about the company. And my email is just Dre, D-R-E, at Ligandal, Ligandal.com.
Thank you very much. And thank you for your time. Back to you, Miriam, to close out.
Thank you. Well, on behalf of Dr. Christine Durbach, who couldn't be with us today, I would really like to thank you, Andre. You did this on very short notice. You're incredibly insightful. It was very informative, and I'm not sure I understood everything you said, but it will definitely be something that I will look into in the future. And I'd like to say I hope I never need your therapy, but and I'm sure that many people will be transformed by it. And thank you so much for a very good, excellent, incisive presentation. Thank you, Susie, for moderating as always. Thank you.
Mariam, for the opportunity, and to WIT and Susie, thank you so much.